Thank you very much. Let's see if I can successfully share my screen as well. Okay. So I'll give you a little bit of background on Wabash College. This is specifically for undergraduate teaching. So rather than talking about the research that I do with undergraduates, I'm going to talk a little bit about how I have transitioned some of that research that's done in my lab to a teaching experience for students who are in organic chemistry too. And Wabash College is in Indianapolis, Indiana. It's very small. It's about a little bit less than 900 students, and all of our organic class comes together. So that means I have chemistry majors, biology majors, pre-health interested students all at the same time. My class size is around 40, and what we do in our class sequence is to introduce NMR early and to try to have students use that spectroscopy to really develop an understanding of how to analyze compounds, not just in the traditional kind of undergraduate curriculum, which is, here's an NMR, tell me what the unknown is, but instead is an analysis tool for all of the experiments that they do throughout the year-long curriculum. I'll go ahead and get started then with some of the presentation content. So what I hope to tell you today is a little bit about the synthesis that we're drawing from the literature, why I think it's great to use in an undergraduate lab, a little bit about the experiment itself, and then some about the analysis. So we have, while we're a small institution and primarily undergraduate, we have a lot of support for instrumentation, and so we have a JEOL 400 megahertz NMR here on site that I take care of and use in our undergraduate labs. Students actually run the samples here as well. So this is the reaction that we're doing, focusing on in this lab, and in the literature, a slightly different reagent conditions were put together, but we were looking at sort of this interesting, as a dye chemist, right, this interesting colored molecule that we made that had all kinds of properties, and really through simple acid catalysis built up this somewhat complex structure. So for the students, I try to kind of simplify things a little bit and show them where it all comes from, right? So 3 equivalents of that starting material end up in the product. And this is a point of access to the students because I introduce it around where we're talking about aldol condensation. So for the literature, I'll show you sort of the mechanistic hypothesis here that starts really with an acid-catalyzed aldol condensation, something that is, just a core part of the curriculum at the time in organic chemistry too. But when these first 2 molecules come together, it's really well set up for an intramolecular reaction that starts this whole cascade and eventually, incorporates 3 molecules of the starting material to get to this more complex product. So for students at the undergraduate level at this point, they can maybe do that first aldol condensation, but seeing how everything comes together is a little bit of a challenge for them. And I think it pushes them in this lab, which is exciting in a lot of ways, for them to see a little acid catalysis can get you some pretty complex molecules. And the molecule that I've shown here is just one sort of structure. Under different conditions, we found in the lab that it's actually really an interesting compound that can have different colors, which you'll see in a few moments here. So I walk through this mechanism with students kind of before they get to the lab, and they try to start to wrap their heads around it, and then they see the reaction itself. So part of why I love this lab is it's a cascade reaction. It's something a little bit more complex than a simple chalcone synthesis that you might see in a traditional organic curriculum. It also has solvent-free conditions. So the reaction's done at high heat, around 100 degrees Celsius, but there's no solvent. And this is one of the first times the students see that. A lot of the organic chemistry reactions involve dissolving something in solvent, stirring it for a while, concentrating it and purifying it, right? But this is a little bit different for them. And so getting that new view is helpful. Again, I'm totally biased, but I'm a dye chemist, and dyes are just fun. So students get excited about seeing colors. I often hear from students, I heard organic was all about white or off-white compounds. While that's generally true, this is an example for students to kind of see something different. In addition, I've taken here an image from the abstract of the original paper in the literature, cited down below from 2019. And so this molecule changes its behavior. Sometimes it can be colorless in certain solvents, and sometimes it's bright red. And as we'll see, there's some other color characteristics as well. So understanding, I think for students, understanding that the local environment affects dyes and molecules more generally, right, in such dramatic ways is a learning outcome for them, something that's a little bit different. And finally, I think the NMR analysis is really interesting, which I'll get to in the talk. So what does the experiment actually look like? We tried to make it as simple as possible so that any lab could do this. You don't have to have special kind of aluminum heating devices or even an oil bath. We just did it in a sand bath and kept it at 100 degrees the whole time. So when it starts, you just have these two white solids that you put together, tosic acid hydrate and the starting material. At 10 minutes, you can already see the melt forming and the color change starting. So, students are starting to get excited. And at 20 minutes, it's getting more intense. And after 30 minutes, we actually stop the reaction. You can see the color is relatively intense. And the other image I show here just shows the consistency. So it is a liquid at this point. And then when it cools down, it gets kind of like a sludge. So a little different than the starting material. But at this point, students are already invested because they see a color change. There must be some reaction happening. Clearly, things are going really well for them, right? They get to the extraction. And this is also pretty fun. So the molecule on the left that I have here shown and the separatory funnel is this bright color. This is just the molecule straight from the vial in about 10 milliliters of dichloromethane. Bright red, and then they quench the reaction with aqueous bicarb, and that gives it a slightly basic environment, changes the structure, and they get this beautiful blue color. So it goes from this kind of intense red or orange through a little bit of purple to this really intense blue. And here's where I usually hear the wows and what happens, and, they start conversations around sort of how that could be possible. So from the literature, these are a couple of suggested states of the molecule. And another added benefit of this lab is I get to ask students, what about these structures might be responsible for color, right? So bringing in a little bit about conjugation in different states, different ways that molecules can be arranged with the same atoms, and acid-base states as well. So this is really fun for them, and then they just continue with the fun. Forgive my my smiley face there, but they run a TLC, but not the traditional TLC to see what's happening in a reaction. What they do is they just spot pattern on a TLC plate and then put it in some ethyl acetate, and they find that it retains the blue color on the silica gel, but when it's in an environment of solvent, something a little bit different happens. It becomes colorless. So this too to them is a little bit perplexing, and it starts a conversation about what might be different in a sort of solvent environment, saturated solvent, versus just the silica plate itself. What is silica gel? What kinds of interactions might encourage different dye forms? And these are the conversations that I love as a professor because it gets students to really dive deeper. So I've shown you some of the fun of doing the experiment. How about the NMR fun? So these really intense colors, they get an NMR back that looks something like this. And you probably can tell before I even walk through it, it's basically starting material. So starts the conversation of, wait a second, I had this really intense color, but what I'm seeing here doesn't match my product. What doesn't match? So there is some residual solvent. We don't put this on a high vac. We're going through the process rather quickly. And it's a beautiful NMR in some ways of the starting material. You can see great splitting there, and I feel like I should put it on an exam someday. But they get kind of frustrated because they're trying to fit, as they typically do, right, a product with this NMR, and it's not working. But in their head, they're also struggling with, well, I saw this big color, why isn't it here? obviously, we've done a little bit more with NMR. It's there, it's just on the baseline. But for a learning gain for them, right, seeing this first and then zooming in to different regions helps them to understand kind of what they're actually measuring here. So I've zoomed into a region and kind of changed my integration a little bit here so that this tight doublet here that shows long range allylic coupling is on the product, this CH3, and the peak that goes right off the page because it's so large is the CH3 in the starting material. So students, certainly this is a region that has other background noise in it, but students can start to see that there are identifiable peaks from the product, and that integration isn't just a ratio between protons within a molecule, but can also be a ratio between protons in two molecules and can help them to get that conversion or yield. So for students who are doing research and those who kind of use NMR frequently, this is a really clear outcome, right? Of course, you could use NMR to see how much you have, but for students who are going through the curriculum and are used to seeing in a book a pure NMR sample, this is something that challenges them and that they have to work through and think about what this actually means. Again, this region isn't beautiful, but there is a pretty great peak that is in the kind of alkene region here that doesn't show long range coupling. So it's the blue proton that I've indicated in the product. And you can see all three of the protons in the starting material here. They're not perfect, but because there is some noise underneath. But you can get a pretty good idea of what the percent conversion or the percent yield is. So students then grapple with, okay, I have an intense color. I clearly have a small amount of material, but now I can actually tell how much I have. And then come up with sort of further experiments, maybe longer time, maybe different conditions, right? What could we do to improve it? As a dye chemist, the ability to use NMR in this way to both prove that a product has been formed, right? But also to suggest that this reaction isn't very far, right? Is powerful as an educator because dye synthesis usually is very high temperatures, very long periods of time, kind of a sludge, and then purification outside of that. If you can recrystallize it, amazing, but otherwise dye synthesis can be challenging. So this is a procedure that's both easy to carry out, it's fun for the students, and some of this analysis tells them something about these types of cascade reactions and how quickly products are actually formed. So these are the NMR tubes that we got from the class, and you should hear conversations that they have there about like, wait, why is this one color and that's another color? The molecule is just really pretty fun to play with. But student learning outcomes, I think they definitely hear that intense color doesn't necessarily mean a lot of dye. And to me as a dye chemist, that's important. You can have a couple molecules and it can light up, right? So that's kind of fun for me to hear that conversation. They learn that those baseline peaks aren't just something to be ignored. Sometimes they're important. You have to consider them. They use crude NMR analysis to see minor products. Again, that's something that that makes sense to me as a synthetic chemist. That's my training, but to them is a little bit new that they can use crude NMR in this way. I mentioned earlier the idea of integration of protons on different structures to tell us how much of each thing is, and maybe an idea of conversion or yield. And I think they see the power of NMR to be able to help us to answer different questions. So just as Edward and the students were talking about in the earlier presentation, there's so much you can answer when you get good QNMR. And for students, often they're learning it just as a spectroscopy technique. This brings it into kind of what else can we analyze as opposed to just proving we made something. Other benefits that I see to the lab generally, great connection to class content. I mentioned the enol chemistry and spectroscopies and conjugation with the color. It brings in ideas of green chemistry because there's so little solvent involved, just that dichloromethane and the extraction. And that solvent-free chemistry actually comes around later because the students do a self-designed synthesis project in my class. And many of the procedures that they find end up being solvent-free reactions, either melts like the one they see here, or even just solid phase grinding. And so having them encounter that in the sort of training before the synthesis project that they develop is really helpful for them to get that experience. Finally, the colors, right? The wow factor, getting students excited at that point in the semester about, a reaction that's pretty complex is good for my teacher soul. And really getting them to understand that the conditions that they study a molecule in can really affect what the structure looks like, and things as drastic as pretty big color changes with different pH and different solvents. So with that, I'll take a moment to thank all of those who worked on this, both in the research lab with me and kind of bringing it into the teaching lab. So Taylor Neal, who did that original work and who continues to collaborate and talk to me about how to make this reaction better and more interesting. The 3 students, undergraduates in my lab who developed some of the reaction conditions: Andrew Jameson, Chukwunolu Chukwuma, Austin Shivington, and the chemistry department here at Wabash College, who's always supportive of me playing in the lab and bringing my research with me. And thank you.
Thank you so much, Laura. It's Mike here. So I'll just interject with a question, and then we'll see if someone else has got a question as well. So first, I wanted to just highlight the really important point you made, that all of the species in the tube have the same response factor. That's a great point, and I think it gets overlooked quite often. So thank you for that. So I actually have 2 questions. One is, so in this case, I guess, in the aromatic region, you've got signals that are quite close to one another, and overlap is potentially a problem. So I was wondering, do you see the alkenic signal that's coupled with the methyl group, long-range coupling there?
Yes, we do see that. That wasn't in the range that I showed,
Right.
but
Okay.
it's because it's coupled, it's a little bit smaller. So that singlet is just a really nice reference peak for them.
Sure. Nice. And then not so much a question, but more kind of a general education. Excuse me. Would you like to say a few words, please, about integrating the signals of tiny peaks accurately so that you can compare them nicely with the really big peaks?
I think that there's some work that I can do there, and that's part of what I want to do in the future. So what I'll say is you saw that my peaks weren't perfectly integrated. They aren't all the same number, and I do have some baseline stuff in there that's shifting them a little bit. I think what students start to see is too, that we often, in a general spectrum, we just round numbers because that's easy to do, whole numbers within a single molecule. So 1 to 1 to 1 could be 1.05 to 1.2. They just sort of naturally round. But here it starts to beg the question, why is it 39 to 40, right? Why isn't it 1 to 1? And so I'm playing with that a little bit for the next iteration of this lab to try to get a little bit better agreement there and more confidence in my integration of the smaller signals. But I think getting students to think about that careful counting, that careful integration is a win for me because, man, it kills me sometimes when they say 1.5, that rounds to 2, right? Like, oh, I'm not sure about that.
Okay.
Thank
Great.
you.
Yeah, thank you. Are there any other questions? I don't see any in the queue. Sorry?
Sorry, I actually had a question. So from my experience with neat reactions, or like ones without solvents, it's very ironically not neat. So I was wondering like what kind of yields you're getting from those reactions.
Yeah, so this is a really low yield reaction for the 30 minutes. If you run it overnight and then recrystallize the product, I think the yield in the literature is somewhere around 67%. But students in my teaching lab just have 3 hours. So in a way, I'm trying to kind of condense condense the experiment for them, and NMR allows me to still get to some of the questions I want them to answer. But yeah, if you run it overnight, you get around 67%. And considering all the steps, I think that's actually pretty good. It crystallizes out really, really nicely. Thank you for that.
Yeah, I see. Thank you.